Welcome back to Biochemistry Learning on the Go for the fifth installment in our series, Chemistry of Amino Acids.
Right.
It's fascinating how these tiny molecules
Yeah.
are really the foundation for the incredible diversity and functionality of proteins.
You can say that again?
And as professors who've spent years teaching this stuff,
Oh, yeah.
I think even we're still blown away by it.
I agree. I sometimes feel like we're just scratching the surface, you
Right.
know? It's easy to get lost in the complexities of large biomolecules,
Yeah.
like proteins, but understanding them starts with those smaller repeating units called monomers.
It's true.
You could say it's like building a house.
No.
You can't do much with just a pile of lumber, but once you understand how those individual pieces fit together, you can build something amazing.
And in the case of proteins, those pieces of lumber are amino acids.
Right.
Let's dive into what makes an amino acid unique, shall we?
Mm-hmm.
Imagine them as tiny Lego blocks,
Okay.
each with a standard base, but a different attachment on top.
Right.
At the molecular level, that base is a central carbon atom,
Uh-huh.
the alpha carbon,
Yeah.
bonded to an amino group, NH2,
Okay.
and a carboxyl group, COOH.
Right.
But it's that unique attachment, the side chain or R group,
Right.
that gives each amino acid its own personality.
Exactly.
Yeah.
So that R group
Yeah.
can be as simple as a single hydrogen atom,
Okay.
like in glycine.
Uh-huh.
Then you have alanine with a slightly more complex methyl group.
Yeah.
And then things get really wild.
Okay.
Like leucine with its bulky,
Nah.
water-hating alkyl group.
Okay, so the R group is like the wild card in each amino acid.
Yeah, you could say that.
What does this variety actually mean for how proteins function?
Well,
I mean, does it really matter if the side chain is a hydrogen atom or a big complex structure?
oh, it matters tremendously.
Really?
The R group determines how an amino acid interacts with others in its environment.
Okay.
Some R groups are hydrophobic, like leucines,
Okay.
meaning they hide from water. Others are hydrophilic and love to mingle with water molecules.
Oh.
Think of it like a party.
Uh,
Some amino acids are the life of the party, drawn to the crowd, that's the water,
right.
while others prefer to hang back in a quieter corner.
I see.
This dance of attraction and repulsion between R groups
Yeah.
is what drives proteins to fold into those intricate 3D shapes,
Okay.
which are absolutely crucial for their function.
Ah, so the side chains are kind of like social butterflies or wallflowers.
Yeah,
Determining how the whole protein party will shape up.
I like that.
It reminds me of how a protein shape determines whether it can act as an enzyme,
Yeah,
a transporter, or even a structural component.
exactly.
It's all about how those amino acids are arranged and how their R groups interact.
Precisely.
Yeah,
And let's not forget
right,
that there are about 20 standard amino acids,
right.
each with its own unique R group and personality.
Okay,
We can group them based on their R group properties:
Ah.
hydrophobic,
okay,
aromatic, basic, acidic, and nucleophilic. For
right.
instance, leucine, as we mentioned, is hydrophobic.
Yeah.
Then you have phenylalanine with its ring structure,
Mhm.
making it aromatic.
Uh,
Lysine with its extra amino group is basic.
yeah,
Aspartic acid with its carboxyl group is acidic.
right.
And serine with its hydroxyl group
Okay,
can act as a nucleophile,
okay.
sort of like a tiny molecular hook that can participate in chemical reactions.
It's amazing how much diversity you can pack into such a small molecule.
It really is.
Speaking of diversity,
Okay.
let's talk about something that our students always seem to struggle with.
Yeah,
Essential versus non-essential amino acids.
right.
What's the deal with that?
It all boils down to what your body can and cannot make on its own.
Okay.
Essential amino acids are those our bodies can't synthesize,
Uh-huh.
so we have to get them through our diet.
Right.
Non-essential ones we
Yeah.
can make ourselves.
Okay.
It's like having a pantry stocked with some ingredients.
Yeah.
But you still need to go to the store for others to cook a complete meal.
Oh, I see.
This is why a balanced diet is so important.
Right.
You need all the essential amino acids to build the proteins your body needs.
That's a great analogy.
Thanks.
So we've got these amino acids, each with its unique personality,
Yeah.
but they don't just float around in our cells randomly.
Right on at all.
They're influenced by their environment,
Right.
particularly the pH.
Absolutely.
And this is where the concept of zwitterions comes in.
Okay.
We often see amino acids drawn in their neutral form,
Uh-huh.
but in reality,
Yeah.
they can exist in three forms:
Okay.
cationic, zwitterionic, and anionic.
Right.
Okay, now you've got to break that down for us.
Sure.
What are these zwitterions you speak of?
Think of an amino acid as a tiny magnet
Okay.
in an acidic solution.
Yeah.
It's like the positive end of the magnet is stronger.
Okay.
So the amino acid becomes positively charged, or cationic, in a basic solution. It's the negative end that dominates, making the amino acid negatively charged,
Okay.
or anionic.
So it's like a tug of war between the positive and negative charges,
Yeah,
with the pH determining which side wins.
that's a great way to put it.
Yeah.
And at physiological pH,
Okay.
which is generally neutral in our bodies,
Yeah.
both charges are present,
Uh-huh.
and that's the zwitterionic form.
Okay.
This ability to shift between forms depending on pH is crucial for amino acids to do their jobs.
Oh.
Imagine trying to build a house.
Yeah.
If you're lumber, the amino acids
Right.
kept changing its shape and charge depending on the weather.
Wow. That would make for a pretty chaotic construction site.
It would.
And speaking of building,
Okay.
we've talked a lot about individual amino acids.
Yeah.
How do they actually link up to form the proteins that carry out so many vital functions?
Well,
I know it involves peptide bonds,
uh-huh.
but could you walk us through that process?
Certainly.
Oh.
It's like making a chain, linking one amino acid to the next.
Okay.
The carboxyl group of one amino acid reacts with the amino group of another,
Right.
forming a strong peptide bond.
Okay.
Imagine a line of people holding hands.
Yeah.
Each person is an amino acid,
Uh-huh.
and their clasped hands represent the peptide bond.
So it's a chain reaction where each amino acid grabs onto the next one,
Right.
forming a longer and longer chain.
Exactly.
And this chain is what we ultimately call a polypeptide.
Yes.
The precursor to a functional protein, right?
Precisely.
Yeah.
The sequence of amino acids in this polypeptide chain,
Okay.
determined by our genetic code,
Yeah.
dictates the protein's ultimate shape and function.
That's incredible.
It is.
But there's obviously more to it than just linking amino acids together.
Oh, yeah.
How does a polypeptide, this long chain of amino acids,
Right.
become a functional three-dimensional protein? That's the real magic, isn't it?
It really is, and that transformation happens through a process called protein folding. We'll dive more into that in the next episode.
All right, I can't wait.
Me neither. Welcome back to BiochemSerye Learning on the Go.
It's exciting to continue our deep dive into the world of amino acids.
Yeah.
We left off talking about how those individual building blocks link up to form polypeptide chains.
Right.
But the story doesn't end there, does it?
Definitely not.
It's like we've laid out all the lumber for our house,
Uh-huh.
but now we need to actually build it.
Yeah.
So how does a polypeptide, which is essentially a long linear chain of amino acids,
Right.
transform into a functional three-dimensional protein? That's the real magic, isn't it?
It really is.
Yeah.
And that transformation happens through a process called protein folding.
Okay.
Imagine taking a long string of beads and twisting and bending it into a specific intricate shape.
Uh-huh.
That's essentially what's happening at the molecular level.
Okay, but it can't be random, right?
Right.
There must be some kind of blueprint that guides this folding process.
You're absolutely right. The information for a protein's 3D structure is encoded in its amino acid sequence.
So it's like each amino acid is a word,
Yeah.
and the sequence of those words tells a story.
Okay.
And that story is the protein's final shape.
I like that analogy.
Yeah.
The specific sequence creates a unique pattern of attractions and repulsions
Okay.
between the amino acid side chains,
Yeah.
guiding the polypeptide to fold into its most stable and functional conformation.
That makes sense.
Yeah.
If those side chains are like magnets, attracting and repelling each other,
Right.
they'd naturally guide the protein to fold in a particular way.
Exactly.
But what forces are actually at play here? We've talked about hydrophobic and hydrophilic interactions.
Right.
But is there more to it?
Oh, there's a whole symphony of interactions happening.
Really?
You've got hydrogen bonding,
Okay.
where slightly positive hydrogen atoms are attracted to slightly negative atoms, like oxygen or nitrogen. It's like
Right.
these tiny Velcro patches
Uh-huh.
holding different parts of the protein together.
So those are the weak bonds we always hear about.
Yeah,
But when you have lots of them, they add up to a pretty strong force.
exactly.
And then there are electrostatic interactions.
Right.
Opposite charges attract.
Uh-huh.
Positively charged amino acid side chains, like lysine,
Okay.
will be drawn to negatively charged ones, like aspartic acid,
Right.
helping to stabilize the protein's folded structure.
Mm-hmm.
I'm starting to picture it. It's like a delicate dance of attraction and repulsion,
Uh-huh.
with all these different forces working together to sculpt the protein into its final form.
That's a great way to visualize it.
And don't forget about the hydrophobic effect.
Oh, right.
Those water-fearing amino acids,
Yeah.
they'll cluster together in the interior of the protein,
Right.
trying to escape the surrounding water molecule.
I don't want to get wet.
So they're like those kids at a pool party who are afraid of getting splashed,
Yeah.
huddled together in the middle while everyone else is having fun in the water. And this hydrophobic collapse helps to drive the protein folding process.
Precisely.
It's a major driving force in shaping the protein's overall structure.
Right.
And then there are van der Waals forces, weak attractions that occur between all atoms when they get really close together.
Yeah.
They might be individually weak, but together they add up to significant stabilization within the protein.
That's right.
Wow. It's truly amazing how all these forces work in concert to guide protein folding.
It really is remarkable.
But what happens if there's a mistake?
Uh-oh.
Can things go wrong in this intricate process?
Unfortunately, yes.
Oh.
Sometimes a protein can't misfold, resulting in a non-functional or even harmful protein.
So what happens to those misfolded proteins? Does the cell just throw them away?
Well, cells are incredibly smart.
Yeah.
They have special proteins called chaperones that act as folding assistants.
So it's like having a team of expert origami folders on hand to make sure everything turns out perfectly.
I like that analogy.
But what if the chaperones can't fix a misfolded protein?
Right.
What happens then?
Well, in those cases, cells have mechanisms to break down and dispose of those misfolded proteins. It's a quality control system to prevent them from causing harm.
It's fascinating how cells have evolved such sophisticated ways to handle protein folding.
I know, right? It's incredible.
But what are the consequences if a misfolded protein escapes this quality control? I mean, can they actually cause problems?
They absolutely can.
Really?
A number of diseases, including Alzheimer's disease, Parkinson's disease, and cystic fibrosis are linked to the accumulation of misfolded proteins.
So those misfolded proteins can actually disrupt normal cellular processes and lead to serious health problems.
Exactly.
Starting to see just how crucial this whole folding process really is.
It is. It's fundamental to life.
It's a testament to the complexity and precision of biological systems.
Absolutely.
And understanding protein folding is not just critical for addressing diseases. It also holds immense potential for designing new proteins with specific functions.
Yeah.
For medical or technological applications.
It's a whole new frontier of scientific exploration.
It sounds like it.
Yeah.
But before we get too carried away with designing new proteins, let's back up a bit and talk about the different levels of protein structure.
All right.
I remember terms like primary, secondary, tertiary, and quaternary structure.
Yeah.
Seemed like a whole new language back when I was a student.
I remember those days too.
Yeah.
But it's actually quite intuitive once you break it down.
Okay.
These terms simply describe the different levels of organization within a protein.
Right.
Think of it like building a house again.
Okay.
The primary structure is like the blueprint. It's the linear sequence of amino acids in the polypeptide chain.
So changing one amino acid in that sequence could change the entire blueprint and the final structure of the protein.
Exactly.
Wow.
Just like changing one letter in a word can completely alter its meaning. Then we have secondary structure, which refers to local folding patterns that arise from interactions between nearby amino acids.
What kind of patterns are we talking about here?
Yeah.
Are there specific shapes that pop up frequently?
There are two main types of secondary structures. Alpha helices and beta sheets.
All right.
Alpha helices are spiral-shaped structures, like a corkscrew. They're stabilized by hydrogen bonds between amino acids spaced 4 residues apart.
I can picture that.
Yeah.
So it's like a delicate spiral staircase held together by those tiny hydrogen bond Velcro patches.
A perfect analogy.
And then we have beta sheets, which are formed when segments of the polypeptide chain line up side by side, like planks of wood, forming a sheet-like structure.
Exactly.
They're also held together by hydrogen bonds between adjacent strands. So alpha helices and beta sheets are like the walls and floors of our protein house.
That's a good way to think about it.
And then we move on to tertiary structure, which refers to the overall three-dimensional shape of a single polypeptide chain.
Exactly.
It's the result of all those interactions we talked about earlier.
Yeah.
Hydrogen bonding, electrostatic interactions, hydrophobic effects, and van der Waals forces.
It's all coming together.
So tertiary structure is like taking those walls and floors and arranging them in space to create the final house.
Precisely.
And finally, some proteins, though not all, have a quaternary structure.
Yeah.
This refers to the arrangement of multiple polypeptide chains or subunits into a functional complex.
Right.
So if the tertiary structure is like a single house, then quaternary structure is like combining multiple houses to form a neighborhood.
Exactly.
A great example is hemoglobin, the protein responsible for carrying oxygen in our blood.
Right.
It's made up of 4 subunits,
Yes.
each with its own tertiary structure,
Right.
all coming together in a specific way to form a functional hemoglobin molecule.
It's amazing how it all works together.
Wow, that's incredible.
It is.
It's like this hierarchical organization from primary to quaternary structure.
Right.
Allows for almost limitless complexity and diversity in protein structure and function.
It truly is a testament to the beauty and elegance of nature's design.
It is.
And as we'll see in our next episode,
Okay.
these different levels of structure are intimately connected to the incredible range of tasks that proteins perform in living organisms.
Welcome back to BiochemSerye Learning on the Go.
It's been a real treat exploring the world of amino acids and proteins with you.
They really have.
From those tiny building blocks to the intricate dance of protein folding.
It's true.
It's amazing to see how much complexity arises from such simple starting materials.
And we've only just scratched the surface.
Yeah.
The real beauty lies in how these proteins, with their diverse shapes and structures,
Right.
carry out such an incredible range of functions in living organisms.
It's like we've built our house, and now it's time to see what we can do with it.
Exactly.
From catalyzing reactions to transporting molecules.
Providing structural support to regulating gene expression.
Yeah.
Proteins are the true workhorses of the cell.
It makes you wonder how anyone could ever find biochemistry boring.
I know, right?
It all comes back to those R groups,
Yeah.
those side chains that we talked about.
Right.
Their specific properties dictate how proteins interact with other molecules and carry out their unique functions.
It's almost as if each protein has its own personality, its own set of skills and talents,
Right.
all thanks to the arrangement of those amino acids.
For example, think about enzymes.
Okay.
Those remarkable proteins that catalyze chemical reactions, their active sites, where the reaction takes place, are precisely shaped to bind to specific molecules like a lock and key.
It's the perfect fit.
And that precise shape is determined by the amino acid sequence and the way the protein folds.
Exactly.
Or consider structural proteins like collagen,
Okay.
which provides strength and support to our tissues.
Its long, fibrous structure, made possible by a repeating sequence of amino acids, is perfectly suited for its role as a molecular scaffold.
And then you have transport proteins like hemoglobin, which carries oxygen throughout our bodies.
Yeah.
Its intricate quaternary structure with 4 subunits working together allows it to bind and release oxygen with incredible efficiency.
It's mind-boggling how these tiny building blocks linked together in a specific order can give rise to such a vast array of protein functions.
It truly highlights the elegance and efficiency of nature's design.
It does.
And as we conclude our deep dive into the chemistry of amino acids, I hope you're left with a sense of wonder and appreciation for the incredible complexity and beauty of the molecular world.
I know I am.
But before we say goodbye, let's test our listeners' knowledge with a few quick questions.
Ready to see what you've learned?
Always up for a challenge, fire away.
All right, question number 1. What is the name of the central carbon atom in an amino acid?
That's an easy one. It's the alpha carbon. Remember, it's the anchor point for the amino group, the carboxyl group, and the all-important side chain.
Question 2. What makes each amino acid unique?
That would be the side chain, or R group, which can vary in size, shape, charge, and chemical properties, giving each amino acid its distinctive personality.
All right, question 3. Name 2 amino acids and describe their side chains.
Let's choose glycine.
Okay.
The simplest amino acid with a side chain consisting of just a single hydrogen atom.
Right.
And for contrast, let's pick cysteine, which has a sulfur-containing thiol group in its side chain. This thiol group allows cysteine to form disulfide bonds, which play a crucial role in stabilizing protein structure.
On to question 4. What is an essential amino acid?
An essential amino acid is one that our bodies cannot synthesize on their own.
Right.
So we must obtain them through our diet.
Okay.
A balanced diet with a variety of protein sources is key to ensuring we get all the essential amino acids we need.
Question 5. What are the 3 forms an amino acid can exist in, depending on pH?
Remember that amino acids have both acidic and basic groups? Depending on the pH of the solution, they can exist as positively charged elations. Negatively charged anions, or zwitterions, which have both positive and negative charges.
Question 6. In a basic solution, which form of an amino acid is favored?
In a basic solution where there are more hydroxide ions, OH.
Right.
The amino acid will tend to lose a proton from its carboxyl group,
Okay.
leading to the negatively charged anionic form.
Question 7. What type of bond links amino acids together in a protein?
That would be the peptide bond, a strong covalent bond formed between the carboxyl group of one amino acid and the amino group of another.
Question 8.
Yeah.
True or false? All amino acids have a nitrogen atom in their structure.
That's true.
Right.
The nitrogen atom is a key part of the amino group, NH2,
Okay.
that gives these molecules their name.
Question 9. What is the significance of the zwitterionic form of an amino acid?
The zwitterionic form, with its balanced positive and negative charges, is the dominant form at physiological pH, the pH typically found in our cells and tissues.
Okay.
This form is crucial for many of the biological roles that amino acids play.
And finally, question 10. Why is understanding the chemistry of amino acids important?
It's important because amino acids are the building blocks of proteins. And proteins are involved in virtually every process that takes place in living organisms.
Right.
From catalyzing reactions to providing structure,
Yep.
transporting molecules to fighting off infections.
Yeah.
Proteins are the true molecular heroes of life.
That's a perfect way to sum it up.
Yeah.
So to all our listeners out there, keep exploring.
Keep learning.
And never underestimate the power of those tiny but mighty amino acids.
Until next time on BiochemSerye Learning on the Go.